set up the electrophoresis box. You will need the gel you just made, an electrophoresis box, and another bottle of buffer. Pour the buffer into the electrophoresis box. Place the gel still in the mold into the electrophoresis box. We have removed the tape from the ends of the gel mold. The gel should be just barely submerged in the buffer. If the buffer conducts the electrical current from one end of the gel to the other, but it will also keep the gel from drying out during the experiment. We don't want to use a dry gel. Alright, we've placed the gel into the electrophoresis box. Next, we're going to be loading our DNA samples into the gel. Here's what we'll need: a loading buffer, the tubes of DNA, the DNA size standard, a micropipetter, the electrophoresis box containing the buffer and the gel, and the pipette tips. So now we're going to zoom in on what we're going to be loading into the wells. We're starting off with our loading buffer. With a clean pipette tip, use a micropipetter to suck up some loading buffer and then add it into the DNA sample. So we've picked up our loading buffer. We're going to drop it into the DNA sample. Next, we'll use a micropipetter to transfer this DNA sample with the loading buffer from the tube into the well of gel. Alright, we're loading it up. First, suck up some DNA samples into the pipette tip. Eject the DNA samples from the pipette tip into the first well of the gel. Nice job! In real life, loading the sample into the well takes some practice, but don't be disappointed if you miss your target in the first few times. Using a clean pipette tip, use a micropipetter to suck up the DNA size standard. Transfer the DNA size standard into the empty well next to it. The DNA size standard contains DNA strands of known lengths. Running it along in the gel will give you a reference by which to estimate the lengths of the DNA strands in your sample. So this is our DNA sample. That was our reference. It's time to turn on the electricity and run your gel. When you turn the power on, the black end will generate a negative charge. The red end will generate a positive charge. Together, they will pass the current through the gel. DNA has a negative charge. To move the DNA through the gel, you must put the black cord, the negative charge, closest to the wells. Because we know similar charges will repel, so the negative DNA will move away from the negative side towards the positive side. Plug the black cord from the electrophoresis box into the matching outlet. And then do the same thing for the red cord. Turn on the power supply. The gel is off and running. Let's look into the gel and see what's happening. Check for tiny air bubbles coming out of the electrodes at both ends of the electrophoresis box. These bubbles are proof that your current is running. Repelled by the negative charge, the DNA moves through the gel towards the positive charge at the other end. Short DNA strands move through the holes in the gel more quickly than long strands. Over time, the shorter DNA strands will migrate farther from the starting point than the longer strands. We can't actually see the migrating DNA the bands that were gray, but we can see the blue dye from the loading buffer as it migrates. So we're seeing that dye as it moves with the DNA. Alright, you finished running your gel. We've taken the gel mold with the gel in it out of the electrophoresis box. The time has come to analyze your sample and estimate the length of the DNA strands. First, you need to stain the DNA in your gel using the DNA staining solution. This stain is a chemical called ethidium bromide. which binds to DNA and shows up under fluorescent light. Although we can't see single DNA strands, we can see larger groups of stained DNA strands. These groups will show up as bands in the gel. Drag the gel out of the mold into the DNA staining solution. Because ethylium bromide stains the gel, it'll take about half an hour to stain. We'll speed the clock up. Remove the gel from staining solution and place it on the ultraviolet light box. So this light box is going to help us see our DNA. Turn on the box. Ultraviolet light from the box can damage your eyes. If you do this in real life, be sure to wear protective gear. Turn it on. Okay, now you can determine the lengths of the DNA strands in your sample. Compare the bands from the DNA sample with the bands from known length from the DNA size standard. So this is our DNA sample on the left side. On the right side is our DNA size standard. Enter the estimated length and base pairs for each band in your DNA sample. Electrophoresis cannot tell us the exact DNA lengths of the DNA strands, just a good estimate. So give it your best guess based on the DNA size standards. Press forward when you are finished to check your answers. Okay, so for this section, you'll be answering this in your Google form.